So thank you so much, Click, for this opportunity. It is always nice to work and learn from so many people here. Excellent talks, Dr. Pratik and Dr. Q. Very nice. This is mostly a repetition. We already had discovered in our sessions. So I'm just quickly going to rush and stop at few points because most of this is covered, and I have restricted myself to the principle of NGS and which work. That was what the title was. So uh, as we have been listening in the morning, that the sequencing is to get their ATGCs right, and that is what we aim to get it and compare with in different disease subtypes. We heard regarding cyber sequencing, and it is an evolution as we are hearing. There is an evolution, but some of the key technologies with respect to sequencing, cyber sequencing, still many times considered as gold standard. Sometimes having problem with some of the essays, you want to go back, sound it and look at because that gives me a lot of confidence whether I'm doing it right or wrong. So sound still forms the backbone of many of the essays, not to be discarded. And we just heard in the morning that you can bring down the sensitivity of Sanger down to 0.1% is what I heard. There is protocol, and we need to discuss that protocol if we are able to use it. Traditionally, Sanger sequencing not to be used for solid or somatic tumors. And whenever you're using Sanger, you please look at your cellularity before you are sequencing that. So you always keep it in mind, when you're using Sanger for your solid tumors. So that is a limitation traditionally. But there are protocols or modifications available as pointed out, but those are not available. You have to see that when your lab is using the protocol, you will be able to get that kind of sensitivity and use that. Subsequently, we have had pyro sequencers being used. Everything is NGS, everything is NGS, and we need to learn, catch up if we are not doing it. Even if we are not doing it, we have to know some of the things. As discussed, again, I'll highlight that NGS has a real advantage over whatever things we are doing. It allows you to look at thousands of genes, whole genes, and everything. So we are moving in that area from NGS to from science sequencing to the NGS, and as more and more automation is coming into this, we are able to get the results even in 24 hours nowadays. So that is what where the field is moving up to. And as you have seen, as shown by Dr. Pratik, there is first generation sequencers which began with Roche, and subsequently we have second generations, and commonly now the second generations include Illumina and Thermo Fisher Scientific, two of the platforms, and third generation Oxford and PacBio means these are long read sequencers, and there are short read sequencers. So second generation Illumina and Thermo primarily are short read sequencers, and the PacBio and Oxford Nanopore are long read sequencers. Even in Illumina, now they are trying to change the chemistry and bringing long read sequencing into their protocols, and whole lot of companies are coming into this space. So this is getting very, very competitive in the times to come. So, and I think this is very important to know. The cost is what drives, many of the things we keep looking at. The whole genome sequencing, the cost of the whole genome sequencing with the type of sequencers we use. It used to be NovaSeq and NovaSeq X, and there we were able to sequence a whole genome by 1,000 teams being made that you can sequence the whole genome at a price of $100 or $200 with the various equipments or various different types of companies in a way. So we are moving in that domain where you can sequence entire genome at a cost of $100, $200. Not at the moment. It is not at the moment, but this is where we are moving. And if you are able to bring down the cost in that way, so you could afford doing whole genome sequencing. So that is where much of the things in sequencing is driven. But yes, there is a lot of scope of targeted panels and the way we want to do it based on our needs and requirements. So there is a two thought processes. Traditionally, clinically, we tend to use in the targeted panel-based testing approach, but then there is also genome sequencing based approach, which is coming down and hitting us now in the clinical space as well. So we are looking at in the spaces. So just wanted to highlight the cost part and the things moving in research domain as well. So these are some of the sequences that we are aware of. Lot of equipments you have Roche, it started with Roche, PGM, solid. Now we have S5, GenXS, and there are a whole lot of sequencers from Illumina. With respect to the Illumina series, you have the Mini-Seq, the iSeq, the MySeq, then the NextSeq, then there is NextSeq 1000, NextSeq 2000, NovaSeq, and now even NovaSeq X, where you can go down to 8 terabyte of data. Traditional 8 terabytes is a huge amount of data to be able to generate. And running that kind of platform needs a lot of multidisciplinary team effort, not only to get the samples, but also to be able to analyze that data, store that data. Huge effort is required when you are running, these kind of services. So traditionally, in a routine clinical lab, it used to be MySeq, and subsequently NextSeq is what traditionally in a lab is what is running. But if you're running few samples of HLA typing, you could even iSeq could be sufficient. So you have to choose your platform based on your needs and requirements. If I'm running an exome or a whole genome, the NextSeq may not be cost effective, or MySeq may not be helpful. I need a platform like NovaSeq or NovaSeq X, and you need an infrastructure or a manpower to be able to do that kind of work and services. So that part you all have to know and understand that while selecting your the sequencer, you need to be able to define your applications and subsequently use for your need. The Illumina platform is, as Dr. Pratik has very nicely highlighted, this is just a revision and other things, we are there. It is the chemistry is the SBS sequencing by synthesis chemistry, and you need to prepare your fragments, and subsequently there is this bridge amplification where you amplify your targets, the DNA targets or inserts that you have, and subsequently sequence it, get the data and analyze the data. But the chemistry behind this is the sequencing by synthesis. And the second platform is the Thermo Fisher, and the S5 prime forms the backbone. So most of the clinical labs are using S5 prime. If you will find that this is the platform that is very, very clinically, it used to be PGM, and it is the S5 from Thermo. And when we talk of clinical space in Indian settings, Thermo Fisher predominates over Illumina. I mean, there are a lot. Primarily, this is what. But now we are seeing some shift in that. Both of them running neck to neck, and even more companies are coming into this space. And the latest equipment is this GenXS platform. Both the systems are available in our laboratory, and we have the experience of running both of them. GenXS is sort of a fully automated system where you just need to load the library, and it tends to give you the results pass depending upon whatever the panels are available. And this is a semiconductor-based sequencing. So it is a pH meter. Any change in the pH that is happening while the sequences are getting inserted, it is detected by this pH sensor, and you have chips here where this the sensors are there, and on this any change is detected by the sensors which are there. So it is a pH meter or semiconductor based sequencing, and that is what you use. So for Illumina, you have sequencing by synthesis. For Thermo Fisher, you have semiconductor based sequencing, and any pH is what is detected. So any I can just share my experience, because this is so sensitive. And sometimes running when we were running at TMC, the water problem, some many water issue could cause problems in this. So you need to look at into small details when you are running that. So small things like water could also affect your experiment if you are not taking care of the pH correctly. And the third generation sequencers, Traditionally, two are there. One is Oxford and Pagden, and they do not have any amplification. This is a single sample preparation instruments, and you have basically long reads here, and even there are two more companies which are coming into this space where so traditionally when you talk of Sanger sequencing, you're looking at around 800 to 1000 base pairs is what you look at when you're sequencing. When you're looking at short read sequencers, it is in the domain of 200 base pairs sequencing. So that is what short read sequencer means. When you are talking about long read sequencer, 20 kb. So one single read is created, and there are any problems or when you're doing an assignment, sometimes the sequence, there is an error or a mutation is present at the end of the read, and that may not be very accurate. When you're taking a call on the read, may not be appropriately covered because the read is not covered. But when you have a long read like this allows you to sequence it very, very correctly, giving more confidence to whatever reads that you have got, but slightly on the expensive side, not commonly available, and some challenges in that area. Like Oxford Nanopore has some error rate. Because of that, you may not be able to pick up entirely the I think there is a discussion of 10% error rate in that. And now, as Dr. K was mentioning, they are mainly looking at fusions, but because large fusions you can easily look at, and that is one area where this nanopore sequencer is looking at into clinical aspects. The PacBio instruments are also being looked at. They do long read sequencing, but running them is very costly. So clinical scale, very difficult to run long read sequencers. So I'll just give you an overview of the different sequencing platforms that are available. Now, all of them, wherever this slide was shown by Dr. Pratik as well, just to highlight that for any of the sequencing that you are going to do, it is the starting material, very important. Whatever template DNA you are going to start, whatever is the experiment, template DNA is good, the rest of the things will be good. And Dr. Spandan is going to talk about how to prepare your DNA correctly. And even before that, pre-analytical variables. If you're doing FFT, what is the fixation? What is the cellularity that has gone into? How is the block? What is the paraffin that you have used? How is the fixation time? Whether it is overfixed or underfixed, they can all affect your template. Whereby any of the experiments might fail, the reads might be less. Sometimes you are able to understand what is the reason. Sometimes you are not able to understand what is the reason. And many of those things will be pre-analytical variables that go into block making. So if you are planning to start this NGS or use it for clinical experiments, please see that your fixation of the block or the extraction material is done in an appropriate way. Traditionally, 24 to 72 hours fixation is what is appropriate. Over fixation, the bone which is there, those cause a lot of issues when you're doing your NGS. And once you got your DNA, then you fragment your DNA because you need fragmentation. And there are different protocols for fragmentation. It could be any of the ways. Then you do your tagging, which is end repair and tagging and subsequently the adapters. This is the sequencer specific adapters are added, and subsequently the amplification happens of the regions that you want to sequence. So this could be by emulsion PCR in thermo, or it could be a bridge amplification in the Illumina. And then you sequence, and subsequently you get the data and you analyze the data. But this workflow remains the same. So you have your DNA, you will have your fragmentation, you will have your tagging adapters, subsequently amplification, and then you have your sequencing, and your data is out. And then you need to look at into your data, how to analyze your data. Making sense? Everyone with me? Or here and there? Come back. And you can ask questions as well. Okay, just to again conceptualize, some of the things that extraction or library preparation is the step number one. Please don't forget the pre-analyticals before that. Even though it is not mentioned here, there are a lot of QC steps involved in the pre-analytical variables. Cellularity being very important. Cellularity or blast percentage. This could be the difference between positive and negative. The blast percentage, if you are not seeing your blasts and sequencing them, you could get false negative. If you're not seeking knowing your TLC before you're running your experiment, you can get your false negative results. If the severity is not right, you can get false negative results, or your math might not be correct. So very important QC part, not to be forgotten, is pre-analytical in anything that you do. Maybe the paraffin that you are using. And subsequently, you have library amplification. And this is, as I mentioned, PCR bridge amplification. You amplify, then sequence, and then you have data analysis. Dr. Tony will be taking that part for you. The extractions, the nucleic acid extraction, as mentioned, there are a lot of QC steps, and Dr. Spandan will be talking in detail in that. Then you have the library preparation, and different types of library is there. It could be a targeted, as Dr. Pratik mentioned, there are different types of libraries you need to prepare. This is very important, the challenges that we are having in library preparation, if you're not doing your QC right, it can destroy your experiment, because when you're pooling, if my library preparation or the library is not of right size, it can spoil your experiment. So after you prepare the library, you need to QC, you see whether the library is prepared right or wrong, and then you do your pooling. Any single library is not right, and you pull it in an experiment, can take the data, and whole experiment can go wrong. Okay, so very important step, that different types of library. Now, as mentioned, pooling is an important part. You pull different types of libraries into one experiment, and if my QC is wrong, my pooling is wrong, my output is going to be wrong. And that is where the common problems or challenges with respect to NGS is there. Okay, so library preparation is a very, very important component. Not only is the panel selection, Dr. Kayur showed you, panels, a lot of panels with leukemia panel, lymphoma panel. That's panels. I can select any panel. These are targeted panels that were shown. I could have an RNA-seq targeted panel. I could have a DNA-seq RNA targeted panel. After preparing the library, I need to pull them together and then sequence it into a sequencer. So for Illumina, there is a library preparation. I think I'll skip some of the slides because this has already been discussed previously. Just I think I'll show you one video where the Illumina sequencing can be highlighted. This is a 3 to 4 minute video.
The Illumina sequencing workflow is composed of 4 basic steps: sample prep, cluster generation, sequencing, and data analysis. There are a number of different ways to prepare samples. All preparation methods add adapters to the ends of the DNA fragments. Through reduced cycle amplification, additional motifs are introduced, such as the sequencing binding site, indices, and regions complementary to the flow cell oligos. Clustering is a process where each fragment molecule is isothermally amplified. The flow cell is a glass slide with lanes. Each lane is a channel coated with elan, composed of 2 types of oligos. Hybridization is enabled by the first of the 2 types of oligos on the surface. This oligo is complementary to the adapter region on one of the fragment strands. A polymerase creates a complement of the hybridized fragment. The double-stranded molecule is denatured, and the original template is washed away. The strands are clonally amplified through bridge amplification. In this process, the strand folds over, and the adapter region hybridizes to the second type of oligo on the flow cell. Polymerases generate the complementary strand, forming a double-stranded bridge. This bridge is denatured, resulting in 2 single-stranded copies of the molecule that are tethered to the flow cell. The process is then repeated over and over and occurs simultaneously for millions of clusters, resulting in clonal amplification of all the fragments. After bridge amplification, the reverse strands are cleaved and washed off, leaving only the forward strands. The 3' ends are blocked to prevent unwanted priming. Sequencing begins with the extension of the first sequencing primer to produce the first read. With each cycle, fluorescently tagged nucleotides compete for addition to the growing chain. Only one is incorporated based on the sequence of the template. After the addition of each nucleotide, the clusters are excited by a light source, and a characteristic fluorescent signal is emitted. This proprietary process is called sequencing by synthesis. The number of cycles determines the length of the read. The emission wavelength, along with the signal intensity, determines the base call. For a given cluster, all identical strands are read simultaneously. Hundreds of millions of clusters are sequenced in a massively parallel process. This image represents a small fraction of the flow cell. After the completion of the first read, the read product is washed away. In this step, the index 1 read primer is introduced and hybridized to the template. The read is generated similar to the first read. After completion of the index read, the read product is washed off and the 3' ends of the template are deprotected. The template now folds over and binds the second oligo on the flow cell. Index 2 is read in the same manner as index 1. Polymerases extend the second flow cell oligo, forming a double-stranded bridge. This double-stranded DNA is then linearized and the 3' ends are blocked. The original forward strand is cleaved off and washed away, leaving only the reverse strand. Read 2 begins with the introduction of the Read 2 sequencing primer. As with Read 1, the sequencing steps are repeated until the desired read length is achieved. The Read 2 product is then washed away. This entire process generates millions of reads, representing all the fragments. Sequences from pooled sample libraries are separated based on the unique indices introduced during the sample preparation. For each sample, reads with similar stretches of base calls are locally clustered. Forward and reverse reads are paired, creating contiguous sequences. These contiguous sequences are aligned back to the reference genome for variant identification. The paired end information is used to resolve ambiguous alignments. Genomic data can be securely transferred, stored, analyzed, and shared in BaseSpace Sequence Hub. Discover the possibilities of next-generation sequencing.
So this was the basic principle and the how the Illumina sequences work. And as I mentioned, there are different types of Illumina sequences: HiSeq, MiniSeq, MySeq. HiSeq is not there now. NovaSeq and NovaSeq X, and they differ with respect to the output that you get. You can have, so depending upon your application, you need to choose your sequencer. We have at the place we have a NovaSeq, and this is there are 4 flow cells on that. This is a very high-end equipment. We are using it for mainly whole exome, clinical exome, and the transcriptome kind of applications. That those are the applications that we are using. There are 4 flow cells. There are SP, S1, S2, and S4. Similarly, on different equipments, there are different flow cells with different varying sizes are there. And SP is around 250 GB data output. S1 is around 1,500 GB. S2 is around 1,000, and S4 is around 3,000 GB output. So that is what some of the sequencers capabilities are there. With respect to the thermo, we have both these equipments. And Genexus is the latest equipment available with us. And the workflow with Genexus, as I showed with Illumina, so many steps are involved in that. With respect to Genexus, the only step that is involved where it is extraction of the sample, and the extraction of the material, and the loading the Genexus with the panels that are available. Okay, there are at the moment 2 or 3 panels available, and there are pre-loaded cartridges. So we load those, extract the nucleic acids, and put it into the sequencer. 15 minutes of hands-on time, and it takes 24 hours for this sequencer to prepare the library, sequence it, get the data out, and subsequently you are able to analyze the data. So around 24 to 36 hours, you are able to get that experiment right. So this is how the current Genexus workflow is. And as I mentioned earlier, this is a semiconductor-based sequencing, and there is an emulsion PCR which is used for the clonal amplification. We won't go into the details; it has already been discussed earlier. For the S5, or the S5, the workflow for the S5 is that you have your extraction, you get your nucleic acids right, do the QC. Subsequently, once you have done the QC, you prepare the library, This is Proflex system that we are using. Once you have prepared the library, do your QC check using tape station or qubit. Then you quantify your library using qubit and real-time PCR, as Dr. Pratik had showed, all these experiments. I'm just putting it in one slide. He had shown everything, whatever we are discussing, like library preparation, subsequently library quantification and pooling, and using your real-time PCR to quantify that. Then you have your emulsion, PCR, and enrichment, and this could either be a manual based using OT2, or it could be an automated using Iron Chef. Different equipments are available here. So either manual or automated, and you sequence. This is the sequencer S5, and there are preloaded cartridges available. Then you get your data, and this is the initial output of the how the chip looks like once the data is out. If there is a problem, this is blue chip. If there is some bubble here, you see here. Anything red means data is fully good, and that's how we tend to on the face value see that this is good output from that. And then the QC steps involved here as well. Once the data is out, so we're going to talk of how the data is looked at. But anyway, this is how the workflow for the S5 thermo is. And this is semiconductor-based sequencing. This is a mini pH meter. There are different chips available here as well. On the Genexus platform, the single chip is available, GX5, and on the S5, there are 5 chips. This is 510, 520, 530, 540, and 550 chips are available. These chips means varying sizes of the data, how much data you can generate, what kind of samples you can pull. That is what this chips helps us. 550 only you can run with only if you have Iron Chef. You cannot, so some combinations are there when you are, you cannot just run each chip like that. Now I'll skip some of them. Third generation we already discussed that there is a and we have discussed in a lot. Sorry. So just a brief, like we saw different sort of equipments, different sorts of technologies, but we need to bring all those technologies into the clinical way. And while I'm bringing into the clinical domain, there are approaches which are available. One is the whole genome approach, where I can sequence everything. As I was mentioning, the cost is coming down, so I can use a whole genome approach. Okay. Then it is the exons. Exome, I could sequence the entire exons. The exome means the target, what the coding regions are sequenced. So out of the whole genome, 1% is the coding regions and 99% is the non-coding regions. So if I just sequence the coding regions, that is what we call it as exome. And there are different types of exomes available. One is targeted exome, or a clinical exome, or a whole exome. Again, within exome also different regions are available. Most of these are in the experimental research phases. So whole genome, exome, traditionally in the research phases. What we clinically do is a targeted panel-based approach, and within that also, it was only targeted DNA-based approach. But we are combining targeting DNA and RNA-based approach together to look at mutations and fusions in one go. How are we doing? So we are doing now targeted DNA and RNA-based combined testing for all these fusions. So all fusions and mutations can be detected in one go. That is what at the moment currently it is being done. And we we were asking questions regarding transcriptome. Transcriptomes mean I am sequencing the entire RNA. This could be mRNA or a part of it. So I'm sequencing entire mRNA to get the information with respect to fusion, with respect to expression, and even other things as well. I can use it for different ways. So whether I can use this is traditionally done in the research mode, but again, because of the sequencing costs coming down, there people are looking at this also into the clinical way. As yet, not clinically validated as yet, but that is also being looked upon. And just to highlight, the targeted DNA and RNA-based approach, you to give you point mutations, indels, copy number variations, fusions, and gene expressions, all can be done. And this is what a targeted DNA and RNA-based testing is being done in the clinical scale. And this also Dr. Pratik showed, you will be hearing amplicon-based or a hybridization based capture. So two different ways of different panels you would be using, an amplicon based or a hybridization based. Amplicon based was traditionally with the Thermo Fisher, but now also available in Lumina. And the hybridization is a non-amplification based, requires more DNA when you are sequencing it. Amplicon based traditionally requires less amount of DNA when you are sequencing using your FFP sample. So these are the two types of panels which are available when you are choosing your assays. This is very, very important. I cannot move ahead. I just thought of including it. Whenever you will be using germline, germline testing, it could be BRCA, it could be exome, it could be hereditary panel. Whenever you are using germline testing, it has to be in the range of 100x. You need to cover that. You heard the comparison being discussed, it has to be 100x or more. The problem comes when you done exome sequencing, the variant that you got is in the range of 15x or 20x. That is where the problem comes. Now, should we believe the variant or not? Is a big question, and traditionally, any variant less than 20x is not to be considered. Traditionally, 20x and we don't consider that variant as a true variant, and there you need sample sequencing to confirm whatever variant that has come, and mostly applicable in the range of the exome or the hereditary panel approach. So the coverage and depth is very, very important, and you cannot escape or not know this. That for germline variants, 100x is what you would be looking at. When you're looking at somatic variants, means you're looking at tumor. You're extracting your DNA from the tumor and then sequencing and looking at mutation. The coverage has to be in the range of 500 or more than 1000x. So minimum 500x, 250 to 500x, and more than that. So it should be more to be able to pick up down to 1% to be that region. For liquid biopsy kind of testing, when you are looking at cell-free, the coverage has to be more because you are looking at 0.1, and then the coverage is 15,000x or 20,000x. So then the coverage is to be more, data is more, the cost is more. So that is why this correlation is there. When you're talking of NGS based MRD, again, the depth has to be 20,000x or more to be again. That also depends on the assay that you are using. But traditionally, whenever you talk about real event detection, your coverage should be more than 10,000x. Anyway, you believe that if you do liquid biopsy at 1,000x, that is not the right way to do liquid biopsy. You will get false negatives. Surely get false negative in that way. Okay. But last two slides, you please be with me. Regarding the bioinformatics. So once you have got the data, so sequence everything out of different ways, you have got your data. So this is raw data that comes out, and subsequently you have your FASTQ files being generated. We call it primary analysis. So any raw data to FASTQ, you call it primary analysis. Usually happens within the equipment. Whatever data comes out. Subsequently, you align it with the reference sequence and create a BAM file, and you have created your variants. You call your variants. This is called the secondary analysis. And from the variants that you got, you generate a report or interpret and generate a report from the different variants. You want to include some, you want to exclude some, you want to analyze that, you want to generate a report, and then this is called the tertiary analysis. So you have in the analysis, primary analysis, secondary analysis, and tertiary analysis. And there are different companies, softwares working in different areas. Just I'm sharing that, sometimes some softwares might take BAM file, some softwares might take raw data, some softwares might take FASTQ files. What data I need to store for my clinical use, so that I can regenerate. Because when you're reporting, you need to store this data for every purpose as well. So all the strategies you need to define based on what data set you need to store and what data set you require for your downstream analysis. This taking in different country, every type of analysis. And this is just to highlight the different softwares which are available. Okay, so there is a genome DNA, SNP, cosmic database, the CBAM portal, My cancer genome. So different softwares are available to help you in your clinical reporting. Traditionally, Cosmic for cancers and clinical for germline are the two commonly used softwares, but there are a whole lot of databases and softwares available which can help you with the tertiary part of doing the analysis part. So with that, I come to the end. Thank you for your patient listening. Any questions, I would be happy to take that.